Well, again, I'd like to thank you for this opportunity. I'm here to talk to you about gemology as an evolving science. I thought what I would do today that might be most helpful is not so much to talk about the types of projects we're working on, but I thought I would focus on more how we do gemological research, how we do studies of gems at GIA. Now, at the end of the talk, you may have some questions, and I don't know if I will have a chance here online to answer them, but if you put any questions to me in writing, email them to me, I'd be glad to answer you in writing. So again, I'm going to try to give you an idea of how we do gemological research at GIA, the techniques we use, and the techniques that we're looking at now and in the future. So gemology is an evolving science, the world of gemology. And I've tried to list some of the ways of that evolution. We have new locality sources of natural gems. There's a wide availability of synthetic and treated gem materials in the market. Standard gem testing with gemological equipment can be challenging. And there's an increasing reliance on gem identification reports. And there's certainly in the laboratory a growing importance of the use of scientific instrumentation for studying gems. There's also a lot of development of gem testing instruments for use by jewelers. And we, of course, have the internet and social media as information sources. Now, if you joined GIA with me in 1982, GI would look very much the same in some ways. We were teaching gemology, but in a research sense, it was very different at that time. We had very limited scientific instrumentation available to us or to anyone else. There were no personal computers, there was no cell phones or email, no internet or social media. Any results we obtained were in the form of paper documents, and we had no searchable information database. Now, of course, 40 some years later, in research, we have an extensive use of analytical instruments. We have scientists and technicians. We have data collection and analysis procedures. We've developed a searchable information database, and we are trying to share our results through publications and presentations as often as possible. I just tried to list here kind of the main areas of research that's going on at GIA. We're looking at all types of gems, diamonds, colored stones, and pearls. And the challenge is identifying those that are natural, those that are grown by a synthesis process, and those that are treated. We're interested in understanding the environments of natural gem formation. We're going to the field to collect colored stones at gem localities. Country of origin determination for colored stones is an important activity for our lab and other labs. We're interested in understanding the cause of color and optical phenomena in gems. And we're involved in the understanding how the cut affects the visual appearance. And finally, testing instrument development. Those are kind of the main areas of research going on at GIA. Since 1934, we have published a professional journal, Gems in Gemology. This is a subscription publication. It comes out 4 times a year. But I wanted to inform your students that if you go to the GIA website, all the issues of our journal from 1934 until just the most recent can be viewed and read and downloaded at no cost. So you can read exactly what we are doing today by going to our website. This idea of an information database is so important. Research on gems requires access to information on the types of gems that you are investigating. And by information, I mean observations, gemological properties, spectroscopy and chemical analysis data, photographs, and other information. That's what I mean by a database. There's certainly a lot of published data on gems, but it's widely scattered in the literature that may be difficult to access, and it may or may not be very useful to the problem that you're dealing with. So we, oftentimes researchers in the gemology field, have to create your own searchable database using your own instrumentation, and whenever possible, obtaining data from gem samples of known origin, that is a known natural occurrence, a known synthesis method, or a known treatment method. These are just kind of the basic crustal environments of gem formation. We have on the left a sapphire in a basalt, which is a magmatic rock. In the center, an emerald and calcite in a hydrothermal vein. In the right, a ruby in a metamorphic rock. And of course, there are a few gems like opal that form in a sedimentary environment. But these primary gem host rocks can weather over a geologic time, and they release gems into the environment, and they can become concentrated in secondary alluvial deposits. And it's the chemical elements in gem minerals, they arise from these geologic environments of formation. Over the past 15 years, we have been going to faraway places to collect rough gems, colored gems in the field, because that provides us information on the local geology, the geographic location coordinates, the sample acquisition conditions. Did we collect the sample ourselves, for example, as well as photographs and video footage that we can use in our educational materials. And since 2008, we've had about 95 expeditions to 21 countries. And we've had now a collection of some 22,000 rough gem samples that are available to us for research. And the photo in the center is just a view in Ethiopia. And I don't know how well you can see it, but there's kind of a dry river channel in the right center, and people are digging sapphires along that river channel. In the colored stone market, there is an importance placed on the country of origin of colored stones, such as, in particular, ruby, sapphire, and emerald. And here I showed us 3 examples of very high quality blue sapphires from different localities. And despite their similar appearance, in the market today, these sapphires would be valued differently because of their geographic origin. Of course, we were not present when these original rough crystals were found, but we're asked, we and other laboratories are oftentimes asked, to give a country of origin report. This report is an expert opinion on the basis of the researchers and the gemologists, based on the gemological features of the gemstone that we're examining. And these gemological features, again, depend on the conditions, environments of gem formation. This is the standard gemological testing equipment that gemologists typically use today: microscope, refractometer, ultraviolet lamp, fluoroscope, dichroscope, spectroscope, a balance, and a leverage gauge for measuring proportions. This is what gemological training GI educates our students to use. But we're now increasingly involved in the using scientific instrumentation, and there are really 3 areas that we focus on for characterizing gems: microscopy, chemical analysis, and spectroscopy. But when you're anticipating or thinking about acquiring a piece of scientific instrumentation to study gems, you have to keep a couple of things in mind. For the most part, you have to be analyzing gems non-destructively. You can't alter the color. You can't cut the gem in half, for example. So it has to be, for the most part, non-destructive analysis because these gems can be very, very valuable. Scientific instrumentation has a significant purchase price and an annual service cost, a contract cost. You need for trained operators and data analysts. You need a kind of a standard data collection and analysis procedure. That's so you can compare the data collected on many different gem samples. You also have to kind of periodically calibrate your instrument to ensure it's operating correctly. And you have to fit the gemstone in the instrument to analyze it. So you may have to modify the sample chamber to hold a gem sample, which could be a gemstone. It could be a gem mounted in a piece of jewelry. It could be a mineral specimen, a crystal, or it could be a carving. You cannot alter the gems to study them unless you own them. And then, of course, you can do what you like. So we're going to start with microscopy. This is probably the most important tool for gemologists. And we, and I'm sure you there, are oftentimes teaching gemology with photographs. So we have a couple of microscopes and we are taking pictures of features in gemstones. I have this picture of this book on the top right. It's by the famous Swiss gemologist Edward Gubelin and my colleague John Cuevla at GIA. And this is kind of a photo atlas of inclusions, and it's kind of a reference book. And it is kind of a picture of a fluid inclusion in a barrel with transmitted polarized light, so you get a lot of different colors, but you see all the different phases in the fluid inclusion. Well, we're photographing inclusions in gems because they are direct evidence of the geologic environment of formation. So just a couple of examples, Cambodian sapphire with a pyrochlore inclusion, a Colombian emerald with a multi-phase inclusion, and a Mozambique ruby with some sulfide mineral inclusions. These inclusions are trapped from the surrounding environment and are important for gem identification and color origin, country of origin determination. Chemistry is the next real area of research effort. And I just show here, this is just a graph of the important 10 elements that make up about 99.8% of the Earth's crust. Obviously, titanium and iron are in those top 10, but other elements, chromium, vanadium, cobalt, copper, nickel, zinc, manganese, are important coloring agents in gems. And then we also find some rare elements, lithium, beryllium, and so on. These are the type of gems that we are analyzing for in the gem samples that we are working on. So doing a chemical analysis of gems, there are a couple of questions that you kind of have to keep in mind. Is this a qualitative chemical analysis or a quantitative analysis? Qualitative, what elements are present? Quantitative, what elements are present and their concentrations? Is this a volume or a spot analysis? Do you need some sample preparation to do the analysis? And is there any potential sample damage, especially on a cut stone? Now, I just kind of use blue sapphire as an example here. In a gemstone, there are major, minor, and trace elements. In this case, the major elements are aluminum and oxygen. Minor elements from about 0.01 to 1%, in this case, titanium and iron, and below about 0.01% roughly, gallium, chromium, vanadium, silicon, others. These minor and trace elements substitute for the major elements in the crystal structure, but they're oftentimes unevenly distributed in the material. And this kind of is important because if you're doing a spot analysis, it's rather difficult to make sure you're analyzing the right location to make the chemical analysis. And again, this minor and trace element chemistry is very important for gem identification and for country of origin reporting. Historically, mineral analyses would be reported in percent, and typically a percent is an oxide, percent iron oxide or percent magnesium oxide. But today, we oftentimes use a unit called parts per million. And I just wanted to relate what a parts per million is to a percent. So just 1% is about 10,000 parts per million. 0.01% is 100 parts per million. This is the range of where trace elements and minor elements, the ones we're analyzing for, occur. And even today, we're kind of chasing after increasingly even smaller concentrations of parts per billion and maybe eventually parts per trillion. This is the world of gem testing in terms of chemistry. If you're going to do a quantitative analysis and determine how much of the elements are present, you need to analyze a standard material of known origin, of known composition. So most of our analytical work on colored stones is on ruby and sapphire. So we have developed a set of synthetic and natural corundum samples for use as standards for the analysis of these important two stones. And by standard, I mean it contains elements of interest at known concentrations. So we analyze the gemstone and we analyze the standard. We know how much is present in the standard, and we can calculate how much of present of the minor elements are in the gemstone. So we had a synthetic corundum crystal produced for us, doped with elements of interest at known concentration levels. And it was very evenly distributed. That was the criteria. And then we would slice up this crystal, and we would use this and our other standards to all our laboratory locations which are involved in this type of research. We all have the same standards. This is vitally important if you're going to do quantitative chemistry for gemstones, to have standards where you know the concentrations that you're concerned with. There's two real techniques that we use for chemistry analysis. The first is called X-ray fluorescence, or XRF. It's a rapid qualitative volume analysis. You have a beam of incident X-rays from the instrument that eject electrons out of the atoms. And then you get a higher level electron dropping down and emitting X-rays as fluorescence. And those X-rays are characteristic of the chemical elements in the material. So the graph at the bottom is just what you'd see on our monitor, on our screen at the instrument. And you see these light blue, these are peaks. So the peaks are at particular energies, which are characteristic of the major, minor, and trace elements in the material. And the intensities, the heights of the peaks, are somewhat related to concentration. But this offers a quick, non-destructive, all you need is a polished flat surface to get a quick idea of particularly the color-causing trace elements that are present in many colored stones. And this is a tourmaline. It's one of these copper tourmalines. It's got manganese, copper, and gallium peaks. But this is kind of where we start in terms of doing a chemical analysis. The more detailed method is called laser ablation mass spectrometry. So in the photo at the top right, the gray box on the right is the laser unit, and the white box on the left is the mass spectrometer. So what you're going to do is you're going to orient the stone and you're going to analyze along the girdle surface. And the picture on the lower right shows the very tiny 50 micron spots. And we normally analyze several locations around the girdle of the stone, trying to pick areas where there's no color zoning in the sapphire or no prominent inclusions. So when the laser fires, it removes small particles of material, and those are carried by means of a flowing gas into a high temperature plasma, and all the atoms in the material are ionized, and you can detect them with the instrument. So the graph on the bottom left is what the operator is looking at when he has the instrument running. So from about 10 to 40 seconds, time is on the bottom axis. From about 10 to 40 seconds, you're going to be collecting a background signal. No laser's not on. When the background is done, you turn the laser on at about 45 seconds, I guess there, and you see all the graphs jump up. You're going to let that burn through the outer surface of the material for a moment. And then you've got about 30 seconds of data collection. And you can detect all the elements, the naturally occurring elements, down to parts per million range, quantitative analysis. You don't need any sample preparation, but you do produce some minor surface damage. But of course, that can be repolished without too much difficulty. And what you end up with from that type of procedure is a plot, such as this one, which is vanadium on the horizontal axis and gallium on the vertical axis. And this, again, are data points in terms of parts per million. And what you're trying to do is compare your unknown, which is the black circle with the red edge, against known samples, which are shown in the various colors. Known other rubies, in this case, from other countries, and you're comparing your unknown stone from your client. And it'd be nice if the data points kind of plotted in different areas of this graph. But you see, there's oftentimes kind of a frequent overlap of data. How do we address that? Well, there are a couple of things we can do. Maybe we can pick different pairs of elements, or we can graph elements 3 at a time on a triangular plot. But there are a lot of these graphs that are produced looking for hopefully a kind of a clear indication of this unknown ruby, where it comes from, compared to rubies of known origin we've also analyzed that are in our database. Well, it gets even more complicated, and this is blue sapphires, similar kind of plot in terms of iron and magnesium and titanium parts per million. It's chaos. It's total overlap. Because why is that? Well, the geology of the gem deposits in various countries is similar. So the trace elements that end up in this case, the sapphire, are similar. Well, we're doing a couple things to try to address this, and one of them is data filtering. So on the left, this is again blue sapphire analyses in terms of gallium and magnesium on the horizontal two-dimensional graph, and vanadium on the vertical axis. And the unknown, we know where it is because we just analyzed it. It's in that mass of colored data points in the left-hand graph. So one technique we're using is to filter out excess data points that are far removed from the location of the unknown. And when we do that, we strip out all this excess data points, which are far away chemically. And this, now you end up getting a much simpler plot on the right-hand side and a much more likely chance of kind of saying, you know, in this case, this blue sapphire is from Sri Lanka in terms of chemistry. We're also been experimenting with more multi-dimensional modern mathematical techniques to kind of plot data and analyze data. So this is just kind of a data set on the left-hand side, a number of samples listed in the right-hand column. And then A, B, C, and D are just any elements. And if you just look at this set of numbers, it's very hard to see any pattern. But what we're trying to use is modern statistical methods, in this case, what's called principal components, but there are other methods, maybe even artificial intelligence, to help us understand how we can address this problem of chemistry and chemical analysis of gems. And now I'd like to talk for a few minutes about color and spectroscopy. Color of any object, any transparent gemstone, arises from three causes. You have the emission spectrum of the light source on the left. You've got the absorption spectrum of the material in the center, and you've got the color sensitivity of the human eye. So you have these three components: the illuminate, the object, and the observer. And if you change any one of them, the color will change. Now, obviously, if you switch gemstones, the absorption spectrum will change and the color changes. But if you change the source of illumination, you produce what in some gemstones is called the Alexandrite effect, where the gemstone has one color in daylight and another color in incandescent light or artificial light, and it's because the emission of the light source is changing. So we spent a lot of time looking at the spectra of gems, and you are familiar with the light spectrum there at the top, with energy increasing toward the violet end of the spectrum and wavelength increasing toward the red end of the spectrum. Most gems are colored by what's called selective absorption. So on the bottom, I have one of these absorption graphs with absorption going upwards, shown on the left-hand side, and transmission increasing downwards. And I show the visible spectrum from about 400 to 700 nanometers, and from about 400 to 650 roughly, or 600, the light is being absorbed as it passes through the gemstone. What is being transmitted is the red colors, and that's what your eye sees, is the pinkish red color of this diamond. So it's the idea that color in most gems is the result of selective absorption or selective transmission of light as the light passes through the gem material. Well, the path length through the gemstone is important. So if you have a greater thickness of material, you have greater absorption and a darker color. Now, if gemstones were flat discs, such as these pieces of plastic on the left-hand side, spectroscopy would be very easy. You have a known path length. You just measure the thickness of this flat disc, and you can directly measure the absorption. But in gemstones, and this is just a model of light going through a diamond, within gemstones, the light oftentimes bounces more than once. And in case of diamonds, it could be many times. So we do not really know the path length. And so we don't have a direct way of measuring the absorption of light passing through gemstones. And that means a lot of the absorption graphs have no scale on the vertical absorption axis. Well, spectroscopy has been used for a long time at GIA. These are some of our early leaders using a spectroscope in the 1930s and 40s. And what we're looking at in spectra, they provide evidence of optical defects in the atomic structure which cause color. So this is kind of a couple of examples on the left. This is a kind of a two-dimensional representation of the carbon atom arrangement in diamonds. And when you have nitrogen symbolized by the N, and a vacancy, which is a missing carbon atom. When you have that type of defect, the diamond will take on a yellow color. Whereas if that defect were absent, it would be colorless. In a similar way, if you have chromium substituting for a few of the aluminum atoms, that causes ruby to have a red color. So it's these optical defects that produces selective absorption or emission of light. This is my colleague, Bob Crowninshield, who was one of the leading gemologists in the world in his day. And in the 1960s, he began really championing the use of the spectroscope for gem identification. And at that time, he produced a whole series of drawings using pencils and maybe some other types of drawing tools to create some drawings of the visible spectrum pattern of all types of gemstones. And we have these patterns in our gemological books today. And I just show you a couple of examples at the bottom, a couple of colored stones, ruby, zircon, and so on. You see the color spectrum, you see 400 to 700 nanometers, and you see a pattern of dark bands. The dark bands are the regions of the spectrum that the gemstone is absorbing. So you have white light coming through, and part of that spectrum is absorbed. You're going to be sharp bands or broad bands. And of course, because you have the wavelength scale, you can, of course, kind of get a pretty good idea where the bands are located, the nanometer positions. But I think most gemologists really are just oftentimes just looking at the pattern of the dark bands, because that's an immediate indication of many cases what the colored stone is. So again, selective absorption, visible spectra, and color. So here's a yellow diamond. Here's my diagram of the yellow diamond, white light entering the diamond, and it has all the colors of the spectrum. And because of the optical defects related to nitrogen, the diamond is absorbing the blue and the violet, mainly those parts of the spectrum, and it transmitting or emitting the other parts which recombine to give the yellow color appearance. So this is how on the top left, that same graph that you'd see in the spectroscope with the dark bands toward the blue end of the spectrum. And right beneath it is the absorption graph, which we would record. And again, absorption is going upward with no scale because we cannot tell the path length, but we can tell the nanometer positions quite accurately. And you can see the peaks on the graph correspond to the dark bands in the spectroscope. So we use an instrument called a spectrophotometer to record spectra. And we do that because we can look at a wider wavelength range than the visible spectrum because of the detector. So you can look out oftentimes into the ultraviolet or the infrared. We can also detect with the detector very weak spectral features that would be difficult for you to see with your eye in the spectroscope. And we can get an accurate, very accurate determination of the nanometer, the peak positions. So a spectrophotometer, if it's a transparent material, this is kind of an absorption transmission spectra. Or if it's an opaque material like a pearl, you can do a reflectance spectrum. But this is the cause of color, and it's important for gem identification. And just again, it's a typical example of sapphire, blue sapphires. Most sapphires either come from a metamorphic rock or a magmatic igneous rock. And you have a spectrum pattern. You can see the spectrum goes way beyond out into the infrared and down into the near ultraviolet. But it's these absorption features, and we pay a lot of attention to these absorption features because they're what's causing the color. You probably are also familiar with polarized light and polarizing filters. Light normally is vibrating in many directions in an orientation that's perpendicular to the direction that the light is traveling. But when it passes through a polarizing filter, the light is constrained to vibrate in one plane. If you have two polarizing filters and they're aligned in the same direction, they will transmit the light. But if you rotate them 90 degrees to one another, the two filters, they will not transmit any light. So polarizer converts unpolarized to linearly polarized light. In a dichroscope, which is what's, I don't have a picture, but a polariscope, a dichroscope's there on the left. This is what is used to separate isotropic gems, things like diamond, garnet, spinel, things that are isotropic, from anisotropic gems, which break light apart and have different refractive indices. So this is a simple example of a tourmaline, which is a uniaxial, one optic axis, two refractive indices. One is along the long direction of the crystal and one is perpendicular to that direction. So if you put some tourmaline crystals and put a polarizing filter over them and you rotate them, you will see the pleochroic colors. So you want to kind of, in an ideal sense, collect these two colors because you have two spectra to really understand the cause of color. And this is a more complicated but important mineral called tanzanite. It's optically biaxial. It's got two optic axes. Here you're going to have two plates, ideally, so you have two of the refractive indices in one plate. But you see that pair there, photos at the bottom, just by rotating the polarizing filter, the profound change in coloration that this mineral has. So we have a little device that we built for optically orienting rough gem material. So we attach it to a post and you can put that post in this device and you have the optics that allow you to orient this little crystal optically. And then you can take it out of this instrument still in the holder and you can put that into a saw and saw parallel to the directions you want. And you can produce either by sawing or polishing a more of a flat plate or something with roughly parallel sides. So I show a zircon there on the right-hand side. And again, when you record the spectrum and the two orientations, you see the two spectrum parallel and perpendicular to the optic axis, and you see the blue and the gray colors. You have to have oriented samples and polarized light to really understand cause of color in gemstones. We also use infrared spectroscopy, which is very valuable for detecting type diamond, type 1 or type 2 diamond and nitrogen content. But we can also detect molecules vibrating in the gem material. So at the top, this is just a jade, and it's got some polymer and wax absorption features. Those are materials that are used to put into the jade to kind of make it look better appearance. At the bottom is an emerald. Natural emeralds oftentimes have water, and there are a couple of water absorption peaks and also some peaks due to oil or resin, which are oftentimes put in emeralds to kind of hide the visibility of the fractures. We can also do luminescence spectroscopy to understand the fluorescence. So colorless light yellow diamonds often have blue fluorescence. So on the left, you can operate this in two ways. You can fix the excitation, which in this case is at 365 nanometers. That peak that goes off the graph, that's the long wave UV wavelength. And then between 400 and 500, you see this broad peak. That is the emission of blue light excited by that 365 nanometer UV incident light. But you can also kind of switch this around. You can also, in this case, fix the emission, which here is going to be at 440 nanometers, which is the high point of the fluorescence. And you can scan the excitation. And you can see exactly what wavelengths in the UV range are exciting that fluorescence. So this allows us to kind of better understand fluorescence in all types of gem materials by this method. Laser Raman spectroscopy is a really important technique. You have a laser that's aligned along the optics of a microscope. It is a very rapid method of doing mineral identification. And just as an example, I show a group of red stones. It just takes a few minutes to record a Raman spectrum. I've even seen at the mineral shows, we have a big one coming up here in a few weeks in Tucson. There are little handheld Raman units that people are using walking around the mineral show. And if you have a database, you can identify minerals quite quickly just by pointing this device at the sample and getting a Raman spectrum. But because you have a microscope, a gemological microscope, what's very important is you can focus a laser on micro features. In this case, this is a multi-phase fluid inclusion in an emerald. And you can capture the spectrum, solid calcite, solid salt, a gas bubble, and the emerald by just analyzing different spots with this laser because it's aligned with the optics of the crosshairs of the microscope. We can also do what's called photoluminescence, which is fluorescence to a laser light. This is very valuable for looking at natural treated and synthetic diamonds. We actually use a range of lasers because they each excite the optical defects in a different part of the spectrum. But again, these defects are a fingerprint as what has happened to the diamond in the earth or during synthesis or during treatment. We also have a way of kind of doing a photoluminescence map. So I show you on the upper left just a PL spectrum, and I've got a couple of features at 880 nanometers circled. You can set this device to only look at that feature, and you can capture that feature in the diamond. And then you can move the location as shown in the upper right across the surface of the diamond. And you can create a intensity map shown at the bottom. So on the left is a fluorescence image of a high pressure, high temperature grown synthetic diamond. As you see an instrument called a diamond view. You see a crosshatch structure, which is a characteristic feature of a synthetic diamond. And on the right, you have a false color PL intensity map of the distribution of the defects, which again correspond very closely to the structure of the diamond. And it's amazing to me that you have a technique that allows you to look at atomic level defects in their distribution. But we've actually kind of now gone one step farther. We are making 3D PL mapping. We've developed, one of my colleagues has developed a new instrument. It's called hyperspectral imaging. So on the left is kind of this little diagram of all these colored cubes representing different defects in a material. So we can kind of scan a point, but we can also not just scan at the surface, but also in depth in the vertical axis. But what this instrument also does is allows us to capture these defects along a line spectrum. So what we were doing a moment ago is just kind of going back and forth, point scanning to collect this image. But now we can go in a line. In this next slide, these are some of the first false color maps of the optical distribution of defects at different levels within a diamond. Hardly anyone in the world has ever seen these kind of pictures before, because I think this instrument is quite unique that allows us to do PL mapping in 3 dimensions in a depth in a gemstone. We also have a scanning electron microscope, very high magnifications. You have an electron beam, it interacts with the material, but you can image changes in topography, composition, and structure. So this is just an example of a diamond plate in different ways that you can use, and we can, kind of use this very high magnification technique to characterize gems. We also do X-ray diffraction, which is a standard method for characterizing all types of materials. X-rays have been known about 110 or 15 years. Elements, the atoms in a crystal structure are aligned typically in planes. And if you have an incident beam of X-rays, at certain angles, that X-ray beam is going to be reflected. So the equation there at the top, the Bragg equation, you have the incident X-rays, the lambda naught, you have the angle, and you can calculate the sine of the angle, and you can solve this equation for the D, which is the spacing of the atoms in the crystal structure. So if you have a powder, or if you're able to rotate the material around, so you're sampling a lot of different directions in the crystal lattice, you can calculate the unit cell parameters and all the D spacings. And this is the standard way that minerals have been identified. It doesn't tell you about chemistry, it tells you about crystal structure. We also have a high temperature furnace where we're able to do heating experiments up to high temperatures, kind of controlled heating on gem materials. And this is just an example of some of my colleagues. We tried taking a synthetic spinel colorless material and diffusing nickel into it and producing this kind of sapphire blue looking color. One of the newer things we're looking at now is looking at isotopes and analyzing isotopes. And we're kind of focusing on the stable isotopes, in particular oxygen. Most natural elements occur in different isotopes. In oxygen 18, these isotopes travel through the geologic and crustal environment in different ways because they have different masses. So you have a way of ejecting atoms and running those through a spectrometer. And the graph on the bottom just shows you the fact that in terms of sapphires from Mogok and Burma, they have different oxygen isotope ranges. I should have mentioned early on, we try to make gem identifications based on as many types of features as possible. That's when it's a safer approach. We've also been doing a little radiocarbon dating for organic materials, in this case, saltwater pearls and shells. These are from an island off the coast of Venezuela in the Caribbean, which was one of the main reasons to draw the explorers from Europe like Christopher Columbus to the Americas. They were looking for these pearls. And we can radioactive carbon-14 date powders from a pearl or a shell to get kind of a formation age. And this is something we've reported on. We've also been starting to look at DNA analysis. I'm not any DNA expert, but if you extract DNA material from an organic material, you can get some indication for a pearl of the species of the pearl, the mollusk species that produce the pearl. So I'm about out of my time here, but just in summary, the acquisition of scientific instrumentation in combination with classical gemological methods has advanced gemstone testing to new levels. But I think it's vital you have proper data collection and interpretation procedures, along with regular instrument calibration. These are essential for getting good results. And we try to rely on a wide variety of techniques, but the most significant challenge that any organization has is going to do gem testing with scientific instruments is this challenge of building your own database of information, typically with your own instruments. And the best samples come from gems of known origin. And I think with that, 1982, 2023, thank you very much.